Hello, I'm Essay, your AI essayist. Please note that all content, audio and video in this presentation are AI generated. As AI can sometimes produce inaccuracies, I encourage you to verify any factual information presented. With that in mind, let's begin.
The tapestry of life is woven with threads of fundamental dependencies, none more critical than the relationship with molecular oxygen. For nearly all metazoan existence, this diatomic gas is the final, indispensable acceptor of electrons in the chain of cellular respiration, the process that liberates the vast majority of energy-sustaining complex organisms. Yet this reliance is a precarious one. The availability of oxygen fluctuates dramatically, from the thin air of higher altitudes to the ischemic core of a growing tumor or the occluded artery of a stroke victim. For decades, a central enigma in physiology persisted: how does the intricate machinery of the cell sense and mount a coherent, adaptive response to the shifting tides of oxygen? The answer, unveiled through three converging streams of brilliant and tenacious investigation by William Kaelin Jr., Sir Peter Ratcliffe, and Gregg Semenza, constitutes one of the most elegant and complete narratives of molecular discovery in modern biology. Their collective work, recognized with the 2019 Nobel Prize in Physiology or Medicine, elucidated a precise, oxygen-dependent molecular switch that governs gene expression, a mechanism of such fundamental importance that it has reshaped our understanding of physiology, pathology, and therapeutic intervention. The intellectual journey begins with a classic physiological question surrounding the hormone erythropoietin, or EPO. It was well established since the work of French physiologist Paul Bert in the latter 19th century that life at high altitude, where oxygen is scarce, prompts the body to increase its production of red blood cells to enhance oxygen-carrying capacity. By the mid-20th century, EPO was identified as the primary hormonal driver of this process, synthesized mainly by the kidneys and stimulating erythropoiesis in the bone marrow. The mystery, however, lay in the sensing mechanism. How did the kidney cells know that the body was hyposic? The prevailing wisdom suggested some form of heme-based sensor, akin to hemoglobin, that might change its conformation with oxygen binding, but concrete evidence was elusive. The sensor and its signaling pathway remained a frustrating black box. It was into this landscape that Gregg Semenza entered, his initial focus aimed squarely at the genetic regulation of the EPO gene itself. He sought the specific cis-regulatory elements, the sequences of DNA near the gene that mediated its response to hypo. Using a strategy of systematic molecular dissection in cultured hepatoma cells, a line known as Hep3b, and in transgenic mice, Semenza's laboratory painstakingly identified a short DNA segment downstream of the EPO gene that was essential for its activation under low oxygen conditions. They christened this sequence the hypoxia response element, or HRE. This was a monumental first step, locating the genetic switch. The next logical and far more challenging question was to find the protein that flipped it. Through a series of laborious biochemical experiments, primarily employing electrophoretic mobility shift assays where the HRE sequence served as a radioactive bait, Semenza's group fished out a nuclear protein complex from hyposic cells, bound specifically to this element. They named this protein hypoxia-inducible factor, or HIF. Further characterization revealed it to be a heterodimer, a complex of two different protein subunits. One was an existing, constitutively expressed protein known as ARND, the aryl hydrocarbon receptor nuclear translocator. The other was a novel, highly unstable subunit whose levels dramatically increased in hypoxia, which they designated hypoxia-inducible factor 1 alpha, or HIF1 alpha. This subunit was the key oxygen-regulated component. Simultaneously, across the Atlantic at the University of Oxford, Peter Ratcliffe's group was pursuing a parallel line of inquiry. Initially also focused on the regulation of EPO in the kidney, Ratcliffe made a critical observation that broadened the entire field. He demonstrated that the oxygen-sensing machinery was present in a vast array of cell types, many of which had no role in producing EPO. This finding transformed the perception of the problem from a specialized renal control system to a general, fundamental mechanism of cellular adaptation, a core component of life's operational code. His laboratory independently identified the same hypoxia-inducible factor and its dependence on the HIF1 alpha subunit, providing powerful corroboration of Semenza's findings. The convergence of these two independent research programs upon the same molecular factor underscored its central importance in the oxygen-sensing pathway. The discovery of HIF Fermi established the identity of the master transcriptional regulator, yet it opened an even more profound mystery. The central puzzle now became the mechanism of HIF1 alpha's regulation. Both Semenza and Ratcliffe observed that while HIF1 alpha protein levels skyrocketed in hyposic cells, the amount of messenger RNA coding for the protein remained largely unchanged. This pointed toward a post-translational mode of regulation. The cell, it appeared, was constantly synthesizing the HIF1 alpha protein, but in the presence of normal oxygen levels, or normoxia, it was being just as rapidly destroyed. Oxygen was somehow flagging the protein for immediate degradation. The question of how molecular oxygen, a relatively inert small molecule, could trigger such a specific biochemical event was completely open. The answer would emerge from a seemingly disconnected area of research, the study of a rare hereditary cancer syndrome. William Kaelin Jr. at the Dana-Farber Cancer Institute was studying the von Hippel-Lindau disease, a genetic disorder predisposing individuals to a variety of tumours, most notably clear cell renal cell carcinoma. The disease is caused by inherited mutation VHL tumor suppressor. Kaelin's pivotal insight came from observing that cancer cells lacking a functional VHL protein behaved as if they were perpetually hypoxic. They overexpressed a suite of genes, including those for angiogenesis like vascular endothelial growth factor or VEGF, even when surrounded by plentiful oxygen. He noticed a striking overlap between the genes upregulated in VHL-deficient cells and the hypoxia-inducible genes being discovered by Semenza and Ratcliffe. In a moment of brilliant scientific synthesis, Kaelin hypothesized that the VHL protein itself might be a core component of the machinery that marks HIF-1 alpha for destruction in the presence of oxygen. A lack of VHL, he proposed, would lead to the stabilization and accumulation of HIF-1 alpha, driving the tumor-promoting gene expression program. This hypothesis elegantly connected the seemingly disparate fields of oxygen sensing and tumor suppression, providing the critical missing link in the path. The race was now on to test this compelling hypothesis. In a flurry of activity, the laboratories of Kaelin and Ratcliffe, now working on the same problem from different starting points, provided the definitive evidence. They demonstrated that the VHL protein is part of a larger complex known as an E3 ubiquitin ligase. This type of complex functions as the cell's targeting system, attaching small protein tags called ubiquitin to substrate proteins, marking them for degradation by the proteasome, the cell's primary waste disposal unit. Their experiments showed that VHL directly binds to a specific region of the HIF-1 alpha protein, but crucially, this interaction only occurred when oxygen was present. When cells were hypoxic, the binding ceased, and HIF-1 alpha was spared. This confirmed Kaelin's intuition and established VHL as the recognition component of the HIF-1 alpha degradation machine. Yet one final, exquisitely subtle piece of the puzzle remained. What was specific, oxygen-dependent modification to HIF-1 alpha that enabled VHL to recognize? The resolution of this final question is a testament to the power of meticulous biochemistry. Both Kaelin's and Ratcliffe's groups converge upon the answer almost simultaneously. They discovered that the signal was a chemical modification, the enzymatic addition of hydroxyl groups, composed of one oxygen and one hydrogen atom, directly onto the HIF-1 alpha protein. This reaction, known as prolyl hydroxylation, was carried out by a family of enzymes which Ratcliffe's group named the prolyl hydroxylase domain or PHE enzymes, later also referred to as Kaelin enzymes. These enzymes utilize molecular oxygen as a co-substrate. Along with iron and alpha-ketoglutarate, they specifically target two proline residues at positions 402 and 564 in the human HIF alpha sequence for hydroxylation. This post-translational modification acts as an irreversible molecular flag. The VHL protein specifically recognizes and binds to this hydroxylated form of HIF-1 alpha, leading to its ubiquitination and swift destruction by the proteasome. In hypoxic conditions, the PHE enzymes lack their essential oxygen substrate and become inactive. Consequently, HIF-1 alpha remains unmodified. It evades recognition by VHL, and its levels rapidly rise. The stabilized protein then translocates to the nucleus, partners with ARNT, binds to HRE sequences across the genome, and activates a sweeping transcriptional program of adaptation. The elegance of the mechanism is breathtaking. The cell possesses enzymes that directly use molecular oxygen to gauge its own availability, translating a simple chemical concentration into a profound and complex biological response. The oxygen sensors were the hydroxylase enzymes themselves. The elucidation of this pathway opened the floodgates to understanding a vast array of physiological and pathological processes. HIF was revealed to be the conductor of a grand cellular orchestra, controlling hundreds of genes that mediate adaptation to hypoxia. These include genes that promote angiogenesis, the formation of new blood vessels, to improve oxygen delivery, genes that rewire metabolism toward glycolysis, allowing for energy production in the absence of oxygen, genes that control cell survival and apoptosis, and, coming full circle, the gene for erythropoietin. This unified framework explained how diverse tissues respond to oxygen deprivation in a coordinated manner. The clinical relevance became immediately apparent. In cancer, solid tumors frequently outgrow their blood supply, creating hypersic microenvironments. These tumors aggressively hijack the HIF pathway to survive, stimulating angiogenesis to feed their growth and altering their metabolism to thrive. Conversely, in conditions like anemia, particularly that associated with chronic kidney disease, the ability to safely activate the HIF pathway to boost endogenous APO production presented a major therapeutic opportunity for ischemic diseases such as myocardial infarction and stroke, where tissue damage is caused by a sudden loss of oxygenated blood flow. Manipulating the HIF system could offer means to protect vulnerable cells. The legacy of these discoveries is profound and multifaceted. The work provided a complete, textbook-level description of a fundamental biological control system, resolving a century-old physiological question with molecular precision. It has since spawned entire fields of research dedicated to exploring the roles of HIF in development, immunology, and metabolism. The therapeutic implications have been realized with remarkable speed. The detailed understanding of the PHE enzymes as the key oxygen sensors led directly to the development of a new class of drugs, PHE inhibitors. By blocking these enzymes, compounds like roxadustat and daprodus that can stabilize HIF-1 alpha even in normoxic conditions, treating the body into mounting a hyposic response. These oral medications have now been approved for the treatment of anemia in patients with chronic kidney disease, offering a powerful alternative to injectable EPO. This represents a direct and tangible clinical translation of basic science discovery, moving from a molecular mechanism to a patient's bedside in less than 2 decades. The work of Kaelin, Ratcliffe, and Semenza stands as a magnificent example of the scientific process. It began with discrete, curiosity-driven questions in disparate fields of gene regulation, general physiology, and cancer biology. Through independent insight, parallel discovery, and ultimate convergence, it yielded a unified theory of oxygen sensing that is both intellectually satisfying in its elegance and immensely practical in its medical applications. It is a powerful reminder that the exploration of life's most fundamental mechanisms often uncovers the very secrets that allow us to preserve it.
We really appreciate you tuning into this AI-powered presentation. Our goal is to push the boundaries of AI in content creation, and your input is invaluable. If there's a particular subject you'd like to see us tackle next, or if you have suggestions on how we could fine-tune our AI's creative prompts, please don't hesitate to leave your ideas in the comments section. We're excited to bring you more unique content very soon.